extensive data collection effort that we had last year to get baseline information on physical, chemical, and biological conditions in the lagoons. I had a team of 4 PhD experts in different aquatic science specialties that helped put together the plan for this data collection effort. One of them was Dave Tomasco, our limnologist. We also had a hydrologist, an aquatic plant specialist, and a fisheries biologist that were very much involved in that. But I would like to give the most credit to the guy on the left in this slide. Josh Bolt lives here in South Lake, and he probably spent about 150 days on the lagoons last year collecting these data. So, why did we collect all these data? The first reason is that we really needed a thorough understanding of these baseline conditions out there and the interactions between the physical, chemical, and biological characteristics. These lagoon ecosystems are pretty complex and variable. There were some previous data collected by TKPOA and others. We've been using those data, but we needed a much more comprehensive understanding of these variables. Secondly, we really needed to see where conditions stood relative to the water quality standards. We also needed to get good baseline information on the communities of fish and benthic macroinvertebrates, or BMI, in the lagoon systems. And all of this information is being used in the EIR, EIS, where we're evaluating the different alternatives for testing aquatic weed control methods. Jumping down into the different components of the study here, the biggest part of the study by far was the water quality and sediment. So we were out there almost every day through a 6-month period. We collected a lot of information on surface and groundwater hydrology. We looked at the physical characteristics with water, such as temperature and turbidity, the chemical characteristics, including dissolved oxygen and pH and others. We collected a lot of information on the nutrient concentrations. And then relative to Laurel's question about aluminum toxicity, we collected sediment samples. We also collected overlying water at those locations, and we conducted what's called an elutriate test, which follows certain protocols for mixing the sediment and the overlying water for a set period of time, letting it settle, and then testing the concentrations of aluminum in the water so that they can be compared against EPA's water quality criteria for aluminum. We also collected samples for chlorophyll analysis, which is an indicator of the phytoplankton, the algae in the water column, and did some measurements of phycocyanin to get an indication of the cyanobacteria concentrations. Our fish and benthic macroinvertebrate surveys, they included pretty extensive methods all over the lagoons. We had bottom trawls in the deeper areas. We had electrofishing transects. We used minnow traps. And for the benthic macroinvertebrates, we had transects where we looked not only at the communities that were right along the shoreline where you can wade in and use a D net, but we also took some samples of the sediments to look at the insect communities in those deeper areas of the lagoons, which really comprise most of the lagoon area. So we got a good characterization of all of that. And then our partner TRC, they had crews that did some wildlife surveys on the terrestrial side and also wetland delineation work. So in this first picture here, you're going to see 13 little yellow dots There's 3 each in the Marina Lagoon and in Lake Talac, and the other 7 are distributed around the main lagoon. So all these are the locations where we conducted the water quality and sediment monitoring through that 6-month period. I'm going to walk over here. On this second one, this shows where we did our biological monitoring. So you can see these yellow lines here in the deeper part of the main lagoon. And so that's where we had the bottom trawl transects. All of the 3 lagoons together were divided up into 10 different sites. And so what we did there was within each site, we did the benthic macroinvertebrate sampling and also the electrofishing and minnow trap work to look at the fish communities. We show it in this figure only here in the far west, site 6. So the pink dots are the benthic invertebrate sampling locations. The little green lines are the electrofishing transects. It would have been a very busy figure if we'd shown those for each of the 10 sites. So we only showed them over here, but they occurred everywhere. Okay, so just kind of an overview of what we did out there. At these 13 locations in all 3 of the lagoons, we had shallow and deep instruments that were installed for continuous 15-minute interval data recording of temperature and pH and dissolved oxygen. So this was really important because there's a 24-hour cycle in how these important water quality constituents vary. So during the daytime when these big aquatic weed beds are producing a lot of oxygen from photosynthesis and also raising the pH, conditions are very different than they are after the sun goes down and photosynthesis stops. Respiration continues, decomposition continues, and your dissolved oxygen drops and your pH is no longer so high in those areas. So we have good, diel characterization now of these water quality constituents. Altogether in the study, we collected about 1.5 million data points. And so that included these measurements, but also laboratory samples that were collected for nutrients and other things. But a lot of those measurement points were these continuous 15-minute interval measurements. In addition to those, we had continuous monitoring of the water levels in each of the 3 lagoons, and we also had a rain gauge out there. There was hardly any rain, but we monitored what we could. So that's what was going on continuously. Twice monthly we got out there and measured the depth of groundwater in piezometer wells that were placed around the perimeter of the different lagoons. And we also did vertical profiles where we measured temperature and dissolved oxygen and pH at 1-foot intervals from the surface down to the bottom. Dave is going to show some of these profiles later, and you'll see how that's pretty important information, particularly when you have stratification in the summer where the denser, colder water stays down near the bottom and the surface layers warm up, and sometimes they're not very well mixed. So you get a lot of accumulation of nutrients down in the bottom layers, different things like that are going on. Then monthly, we got out and sampled the lagoons, lagoon water for nutrients, total phosphorus, total nitrogen. Several times during the season, we sampled the piezometers for the groundwater nutrients and also measured turbidity in the lagoon water. And then once per year, just in June, is when we had the sediment sampling that was sent to the lab to characterize for nutrient concentrations and also for those aluminum elutriate tests that I mentioned. And June is also when we had our benthic invertebrate surveys and the the fish surveys. I should have said July. It was actually July when we did those. July is also when TRC did the terrestrial and the wetland surveys. So Dave is going to tell us now more about how some of these data were used, and particularly his focus on the nutrient sources and the nutrient cycling within the lagoons.